The world around us is complicated and ever-changing. We must digest this constant stream of information and use it to determine which actions to perform next. Our brains use a shortcut to solve this problem quickly, like when we look at these damselflies hovering above the water. Before we know what they are, our brains have detected each shape and its orientation, color, and aspects of its motion. This allows specialized information to be collected rapidly at the same time, a strategy called parallel processing. But detecting features separately means the information must then be recombined to guide behavior. How does this recombination occur? How can detection of different features in the world actually change our actions?
In this paper, we examined how detected features are translated into actions by studying the brain of the fly, Drosophila melanogaster. We uncovered how the fly brain recombines two separate visual features of a looming predator, such as a damselfly, in order to determine what kind of escape takeoff to do, a long one or a short one. A fly's long and short takeoffs both occur faster than the blink of an eye, but they're really quite different. In a long takeoff, the fly first extends its wings, then elegantly jumps off the ground while starting to flap. In a short takeoff, the fly just jumps. With folded wings, the fly tumbles in the air and must spend time and energy righting itself. So, if the predator is slow, flies more often choose the long takeoff, since wings provide stability. If the predator attack is fast, there's a greater probability the fly will use a short takeoff to escape in time.
Our previous work showed that short takeoffs occur if a single pair of large descending neurons, called the giant fibers, fire a spike before other descending neurons that control long takeoffs. In this study, we asked, what visual features are transmitted to the giant fibers when a predator attacks? And how does integration of these features dictate when the giant fibers spike, and so ultimately determine how the fly behaves?
We recorded giant fiber activity intracellularly while showing the fly looming stimuli that simulate a predator, and using genetic tools targeted to candidate feature encoding neurons in order to block their neural transmission. Initially, we expected to find a single type of neuron providing looming information to the giant fibers. However, to our surprise, we found one type of neuron specialized to encode looming angular velocity, and we found evidence for a second neural type that encodes the looming angular size.
Together with our previous findings, our new study suggests a model in which both angular velocity and size features are summed to determine giant fiber spike time and bias the fly's behavior. Here's how we hypothesize it works. If a predator is attacking slowly, angular velocity is minimal, and its looming profile mostly activates the size encoding neurons, which then excite the giant fibers, but not always enough to reach spiking threshold. Meanwhile, other descending pathways coordinate a long takeoff. When a predator attack is fast, then both size and velocity feature channels are excited, activating the giant fiber more strongly and increasing the likelihood that it spikes early, causing a short takeoff. This parallel processing lets the fly detect an attack quickly by simultaneously processing distinct features of a looming predator, and then combine multiple streams of information to make the right escape choice, and live to flap another day.